centimeters apart and are used for measuring.
Saw the biggest squat lobster, I think.
Saw a
At least us
ginormous.
back here saw.
Yeah,
I've never
squat
seen.
lobster.
It was like 4 inches long.
Yeah, we've been collecting core samples of the sediments, the worms, sucked up some worms off the whale bone.
We have some fluid.
We have some fluid.
And nice photo mosaics.
We got ourselves a squat lobster off the site. We're having a whale of a time. Wah, wah, wah.
Just that guy. Just, oh, no, he's moving.
No, it is not a bad way to close the season.
I was just thinking that, yeah, that's awesome for a last dive, isn't it?
Yeah.
I feel very lucky. Last year to discover the octopus garden, and then this year we did all that great octopus diving, and then on dive 2 to find this. So exciting.
Pretty awesome for any dive.
An embarrassment of riches on my end. The lateral line on those grenadiers is so interesting looking, isn't it? So that's the line that's running down the side of that fish. Doesn't look continuous, so.
Is there some like kind of advantage that line gives the organisms for tricking prey or predators?
The lateral line helps the fish sense pressure changes in the water around them.
Whoa.
Yeah, so sometimes you wonder how if you're approaching a fish, say, from behind, and you put your hand in the water and then they
And it
like
just swim
leave,
away.
and you're like, there's no way they saw me. Well, they felt you.
I always assumed
felt
it
the
was
water
just
move.
the waves, yeah.
Yeah, but
And
that's how they're one of the ways they're detecting
Does it
it.
have like extra sensory?
Yeah, they're pores that can sense the change in.
And they're like they're fluid-filled canals, and they're connected to the nervous system.
Kind of like a little ear canal, like we have?
Yeah, we have. And it also helps them school, because, you know, they don't run into each other when they're in these giant schools.
Yeah, that is absolutely amazing when fish all seem to like, hundreds of fish will shift together.
Can we push in on some of the worms? I remember that it was
Imagine.
mentioned that sometimes they're also bone-eating snails, and we've yet to see that.
Sure.
But maybe they're hiding. Just push in on the jawbone there, where you see some of the worms.
Run lasers on for this as well.
Nice. Yeah, actually, should we turn lasers on to get a good shot of the size of those?
So lasers are on. We can just come a little wide.
Yeah,
There you go.
Oh, can you zoom in on that?
With a little
Never mind.
amphipods, maybe?
Yeah, maybe that was what it was.
Where are you at?
Anyone seeing any little
right there,
snails?
right in the center.
that
zoom
that
in there.
okay.
It looks
Pushing
like
in.
a little amphipod, I think.
Yeah,
right?
it's got a little
That's
white
full
tip to
zoom.
it. Oh, okay.
Yeah.
Thank you.
Okay.
Yeah, I think it's an amphipod. I'm not seeing yet any snails. Maybe over onto the bone. Right now we're really getting a good look at some of the baleen.
Beautiful.
Okay, I'm gonna come wide a bit, looking for snails.
How big would these snails be approximately?
That's what I don't know.
Hmm.
Is that 3 different types of fish in a row? Do we have a grenadier and then a eel pout and then
I think the
I think
rest
they're
are eel
two eel
pouts.
both eel pouts? Okay.
Yeah.
You want to push in a little bit more there, Wayne?
Yeah, I can push in.
Move over bone-eating worm. The bone-eating snail is taking over the deep. Here we go.
Not this deep.
That's full zoom.
Roger. Not seeing much on these bones here.
Let's see. They've been seen, you know, at 2,900 meters, so definitely within the depth zone that we're in. They're little, they're snails that have a spiral shell. But how big are they? Oh, they're pretty good size. I'm
yeah.
seeing them here. They're kind of on scale with some of the smaller squat lobster. So, no,
Maybe.
So they should
Well,
be easy to.
they should be easy to
see.
spot. It looks like they're down kind of in this one picture and amongst the sediments along the margins.
Do they say at what point in the like whale decaying succession that they typically occur?
It was interesting though, that area around this carcass has such a thick carpet of worms. I wonder if that's obscuring our ability to see them if they're there.
Are they sometimes orange? When we were collecting the worms, we were off the sediment. There was an orange thing below where we had collected.
They look more like a tan color. I don't obviously see them.
You guys want a side angle of the jaw again, or?
Can we go to the other side, the far side?
Yeah, sure.
Okay, coming wide.
That's good there, Wayne.
Okay. All right, I'm gonna hand the video over back to Ashley, and we'll see you guys on the 48.
Great.
Bye, Wayne. Thanks.
Okay.
Good seeing you.
Can we zoom in on those two wriggly little
Hagfish?
Eel pouch?
yeah, in that hole? Are they? I wonder if we can get an idea. Are there
Well-
mouths? Are they chomping?
Well, earlier we saw
Ashley, you mind
a
zooming in there,
larger
please?
eel pout in there, and it did a full
Go
like
ahead.
circle
Zoom in.
of its
Uh-huh.
body, like it was
To
ripping off
break off a
some
piece.
some
We're
flesh.
gonna go for that little hole.
Like
of
the
worm-
alligator
of,
spin move.
Mm-hmm. So my guess is they're in there, eating.
I want to see them rip off some of that flesh. Okay, they stopped moving.
They're chewing completely before swallowing.
Jennifer,
Thanks for that zoom.
one of our explorers wants to know
Alright, go
why
ahead
we're
and come a
not
little
seeing
way.
any
Look at this guy.
hagfish.
Oh,
That's a great question.
yeah. That is a good question. What?
Is this too deep for
Well, that's what I'm
hagfish?
thinking.
Yeah.
Yeah.
Maybe I'll have to look up the depth range of hagfish.
Do you guys want lasers on for these shots, or?
I don't think we need lasers on for these shots.
Cool.
No, thank you.
No, I think we're mostly just looking for the snails and
Yes, if we can come around, I think we should look between the margins of the bones and the sediment. Just had been really looking for snails.
But the space is pretty filled with oxidex. Osidex.
I know, he's
Yeah.
just having a
Yeah.
day.
I think we might be too deep for hagfish.
How?
Okay.
I'm
What's their
looking on
depth?
the
Kind of close.
Department of Fish and Wildlife, California Department of Fish and Wildlife website for Pacific hagfish. And I'll let you know exactly what it says. Pacific hagfish are typically found in waters at depth of 100 to 500 meters.
Yeah.
Oh, yeah.
So, Mike, you lucked out. We are too deep for hagfish.
Yes.
Otherwise, this would probably be crawling with hagfish.
Yeah, I imagine. Yeah.
Then we get the
And then you would
octopus.
not be having stress dreams. You'd be having hagfish.
God.
Nightmare.
Imagine
Nightmares.
having hagfish nightmares.
There's
Terrible.
nothing worse than taking a benthic crab and having it come up with a hagfish
Ooh.
dangling out.
But it is interesting. Earlier, someone noted that the octopus may not be eating the actual whale fall, but instead eating the crustaceans associated with the whale fall, like those squat lobster. If we found some of these snails, it's possible they would also eat those, because octopus are known to eat gastropods as well.
Maybe that's why we don't find those snails,
Oh.
because there are so many octopus at the site.
Yeah.
Yeah, the news flash went out to the snail community that they're going to be predated.
Can we put the lasers on and focus in on those tubes that are part of the arteries? It's over to the right.
Over to the right.
Yeah.
These guys up here?
Just a little more to the right, right next to that fish. Yeah.
Sure.
He's behind him now. He's obscuring that view.
The little small ones, you mean?
Yeah.
Yeah.
And they're huge.
Oh, sorry about the visuals.
It's okay. That's actually great. I was able to see. Thank you for that zoom.
Yeah, we'll get you back over to the arteries here.
And so what about that big sort of trumpet-shaped, That thing there we see just in the top that
Yeah,
is round. Is that
I was
like
thinking that was also one.
one? Okay. Or
Yeah.
intestine or esophagus?
It's a similar color as the others. And
Yeah.
it's definitely tubular in the right place.
Mm-hmm.
Well, with respect to the other tubes we see.
Right, right.
You want to come a little wider, video? Pull wide, please, actually, for a minute.
You need.
Got myself all mixed up here.
We've covered all the video requests and sampling requests except for grabbing that one loose bone with the worms on it. So we're going to go ahead and grab that, and then we're going to get going.
Great.
Getting a ball.
I lost track of which side we're on. Are we on the correct?
I think
Oh, there
that's
it
it.
is. Right
This is the
there we
right
go.
side.
Perfect. There they are.
It's like we were waiting for it or something.
We were
Well,
just getting all lined up.
It
What
was good that
about-
we did that photo mosaic with everything intact,
Yeah.
right? Because we
So-
don't want to disturb the site.
So Chad, we were just searching for that whale-eating snail, and we don't see any. One interesting idea is that it could be that they would be octopus food.
Janet wrote back and she said that they haven't really seen the octopuses on whale falls. They've seen them near, but not on, and that she speculates that perhaps rather than eating the ossidex or the whale, they're actually eating the crustaceans.
Hey.
But if they're gastropods, I don't see why they
know.
wouldn't possibly eat them too. We don't know. Yeah. So anyway, just pure speculation, but kind of fun to think about.
So these two that are kind of in front of us here, those are the ones that you guys still want, right?
Right.
Great idea. Actually,
Sign them down. I guess the relief isn't very good. I can reposition us if you want, Mike.
Magda, this sample will be, is it 034?
034, yes.
Am I doing a good job keeping up?
Yeah, absolutely. I think we could probably put it in one of the starboard, like either E or F, one of the larger ones. Probably E, if it's easier to get to.
You guys want the one on the left or the right?
Can we just peek and see which one that has more cover of worms? And I want to go for the one with more.
probably the one on the left.
Looks like the one on the left.
Left, yeah. Go
If
ahead
you
and
can
zoom
see in
in.
the bubble cam, might be better.
That's a good point.
Yeah. Okay. But we do want to get a zoom in and with the lasers.
You racked out all the way?
please.
Yeah, I'm racked out full. Oh, nope, never mind.
There
Just
you go.
kidding. Go ahead and zoom in their video. With the lasers on it as well.
Yes, please.
You know what? Are those worms attached
come
to
a
the
little
whale
wide.
fall above it?
It's good.
Well, so there's definitely some worms on this bone. But I think the one on the right might have.
Is there one to the left
Yeah,
of the
the
one
one
on the left that's standing upright?
That one?
Yeah. Is that?
Might be
Can't tell if
attached. We
that's
could try it.
loose. Let's just
It looks like
kinda
it's got a nice tough.
try and wiggle it.
Yeah.
See if it wiggles loose.
So the leftmost one that's like stood upright?
Yeah, that seems to have the most
Yep,
coverage.
I'll check it out.
All right, you want to come a little wide there, video? That's good.
Zoom in there, video. A little less, sorry. Come back. It's good.
Oh, the lack of snails might also be depth related.
Oh, nice.
The specimens Ruby
I'm gonna bump up a
Spira
little bit there,
were
Mike.
found on an
What's that?
experimental whale fall in
Gonna bump up a
1,000
little.
meters.
But I thought
You're
I
gonna
just
what?
read
You want me to
something
bump up
about
with the camera so
that
you
they
can see
were-
your joints?
Yeah, sure, a little bit. It's okay.
Sorry, throwing you off.
So this one?
Yeah.
Was it loose?
Yep.
Yeah.
Okay, great.
Should I show me the arm for a minute? Full wide. What's going on
A
with
little out
that?
of reach.
No, I just touched it. Should be fine. It's getting there though. Okay, zoom in again.
Oh, look at that. Wow.
Are we
Oh.
sure that's bone? Yeah.
The top part is bone. It's got all kinds of good stuff on
Oh,
it.
wow,
Okay.
yeah.
It's pretty decomposed, it looks.
Do you think it
looks
has
good.
soft tissue?
Yeah, I think so.
Maybe. But, I mean, that's a good amount of worms.
Yeah.
So it's on both sides. That looks good to me.
That's awesome.
Okay.
That's going to be a smelly
grab.
sample.
Yeah.
That can go in starboard E.
Okay.
Okay, full wide, please.
Cool. An awesome sample to process.
Yeah, if you want to help Chess, you're welcome to join us in the wet lab.
All right, switching over.
Aaron, as soon as we get the sample tucked away, I think we'll be ready to
And did
start
we say E,
making-
sorry?
Yes, E.
Okay.
making way to our new location.
Yep.
Okay,
Once
awesome.
we get it settled away, we'll just review the standing orders and get on our
A
way.
little more
Okay,
out, Mike.
great.
There you
Oh,
go.
that's great.
Get the next watch set up for a blue water transit.
Oh,
I don't think it's
said
gonna be
like
blue
a true
water.
professional.
I don't think it's going to be blue water. It's going,
Snarky.
it's going to be. It's not going to be blue water.
Awesome.
It's going to be rapid survey
cool.
of the seafloor.
Yes.
That's right.
Okay.
Rapid
That's right.
survey of the seafloor.
And the water column.
Okay, and that one in starboard.
Starboard E, biobox E. And Aaron, that was sample 034.
034, roger.
Yes.
And
Good job, guys.
looking good. All right. Wow. We did so much good science here. So exciting.
No.
Take off the top and
Yeah.
it'd be cool to take off the top of the vertebra.
So we're not taking one of the big ribs?
Yeah. Are you going to carry it?
Yeah? Stick it somewhere?
No, I think we're good.
Really?
Good we've been told that was a good piece of bone.
Sure.
Yeah, we collected 11 samples.
Nice.
So we have plenty of, data.
And we've gotten, Magda, just so you know, we've got some instructions from different researchers how they want them preserved.
Yes, I made note of that
Okay.
whenever I saw that in the chat.
Okay, great. And then Chad has some other as
Yeah,
well.
like slicing through the bone and that sort of thing.
All right, ROV, let me know when you're
No, it's
ready
more
to
if they
talk
want
about
it frozen,
plan.
formalin,
let's talk
or
about it.
ethanol.
All right, let's talk about it. So
Let's
we're
hear the
here.
plan.
Well, there was
We
one
wanna-
who wanted stuff in.
Just do a kind of a drive-by
Yeah.
on this, and then up to somewhere at the base of the center cone, no real preference, and then they're going to be marching it up. But this is kind of where we're going, so let's just zoom in a bit on just these two.
While we're setting a plan for our transit away from the whale fall towards this interesting cone feature on the
So
seafloor.
saying we want Argus there-ish, maybe there-ish. Does
I
that sound
encourage you
good?
to look at
Sounds
the
good.
Argus
092.
view.
092, roger. And how long is that? It's like a kilometer?
The Argus view is pretty neat right
1.1
now.
kilometers, yeah.
Okay. Sounds good.
Sorry, oh, sorry.
So we're heading
You
to
almost hit
waypoint 16.
Yeah, about there.
But okay.
Yep.
It's for
Go
science.
full wide on
It's
Argus, please.
And just so it's clear, there's no intention to stop there. We're just probably going to turn the corner there, get a kind of drive-by shot, and then be headed to the
Thank
cinder
you.
cone.
Daily candy fix.
Oh, thank you.
That sounds great. All right, everybody, let's say goodbye to our whale friend.
We can fix up our 6
Some
8
great
Bye.
parting,
Awesome.
Yeah, I'll
some
let you
great
get
parting
get
shots
a
in
little bit
Argus.
closer
Rest
to
in peace,
Argus.
buddy.
We'll take that out.
Give me a shout when you guys are ready.
Yeah.
Thanks for sharing with us.
I just drive over this actually instead of doing a pirouette.
Dun, 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 dun.
Well, this is gonna be our last watch, but it was a great watch.
No, we saw so many great stuff in just these 2 dives.
Yeah.
So we're making for the cone now.
Well, we're
I'm
heading
just going to
that
pull
way.
up a bit,
Yep.
Jess, and I'm going to
We're
spin
gonna
it out.
turn a corner by another kind of high relief looking feature on our way to the cone. But that will
Long
be
way.
not on our watch. You see there on the seafloor a number of the squat lobster.
Aaron, the heading is about 90 degrees.
Yep, about that. And just before we get going, Rennie did warn us there's a pinnacle that pops up around here. They didn't have any problem going over it at speed, so we didn't anticipate it being an issue, but just be aware.
Right.
Jess, you're going to have to turn with Argus.
These are, maybe all these eel pouts are just resting after they've filled their bellies. Because they're all just laying on the seafloor.
Yeah, I mean, I can imagine they don't like come across this plethora of food so much, so. You think they overeat?
I think they gorge, yeah.
A
Oh,
lot.
another
They're
really big
huge.
squat lobster there.
Oh, could be the same one.
Could be. Maybe slowly making his way.
What are we doing?
Positioning.
Taking out a wrap that we were delivered with at the beginning of our watch.
They're untwisting their tether.
Tether.
Got it.
Right.
All right, looks good.
So the tether between Hercules and Argus was twisted and you just untwisted it? Is that right?
Yep.
All right.
Someone in their excitement upon finding a whale spun around in a circle.
That's understandable,
Yeah,
huh?
I guess.
And then we got to spend so much good time with that whale. I think it makes sense that we're going to help get everyone set up.
All right, bridge nav.
This is good.
Come down again.
Yeah, go ahead.
Can we start tracking a line bearing 092, 0.4 knots, please?
We just got a hello from Bryce C., Nautilus Ocean Science Intern 2017. Hey, Bryce.
Hey, Bryce.
Hey, Bryce.
Hey, Bryce. So good to hear from you. I had fun sailing with Bryce before, so good to hear from you, Bryce.
Yeah, I sailed with her last year
Mm-hmm.
on the Channel Islands.
I don't know Bryce, but hi Bryce.
Bryce is now in graduate school. That's why she's not out here. Well, hopefully she didn't log on just right now and just miss the whale fall.
YouTube.
That's right. Or rewind the live stream.
Someone asked, do we really have to go?
Oh, yes, sorry, we really have to go, but just, you know, there's still 2 more watches to go, and there could be something really exciting.
There could be discharging fluid.
See, now I don't know if everyone's going to think that that sounds as exciting.
Yeah, but when
Well,
they start to really learn about crustal aquifers
Oh my
and
gosh.
subsurface
Oh,
flow,
behave.
they're going to get into it.
all right, so you have an opportunity right now to just grab them before our watch ends and get them excited for the potential. Why is that so exciting?
Well, it's really exciting because it influences how heat's distributed through the Earth.
Uh-huh. And?
And
Let's keep going with that. Let's keep going.
well, right now it's a really big area of research because they're trying to understand microbial life in the subsurface. And so by looking at the pore fluid as it moves through the crustal aquifer, they can try to understand the different biology that might be related to that flow and the different chemical reactions that are happening as seawater ascends into the cracks that are in the basalt on the bottom of the seafloor and then moves between a recharge zone, which is where it enters, and a discharge zone where it exits, which is what the Davidson Seamount Management Zone seems to be hosting, is a discharge site.
So the squeeze samples that we took on the cruise prior to this one, are they going to help us better understand what's going on with that?
Yes, they should.
How?
We are going to measure them for the chemistry, so we're going to look at how the geochemistry altered itself from bottom water, so we'll compare it to bottom water samples. We're also able to compare it to other sites, so the Juan de Fuca Ridge and the Dorado Outcrop, which are the only two other known
Go ahead and zoom on it.
discharge sites at low temperature hydrothermal vent systems. And we can also look at the biology.
You know, last year when we were doing the Loihi
What
the
is that?
subsea
It's
expedition
downstairs.
at Loihi Seamount in Hawaii, we did find some venting sites, and I mean they were low temperature, but maybe not to like 10 degrees or so.
No, but
we
if you
did
have the
find-
shimmering.
Yeah, we did. Yeah, we did find some, maybe not to the extent that you find here, but
Yeah, I mean, I
Could
feel
that
like
be a sea
abnormal-
urchin
yeah.
test? Maybe?
Yeah.
learning
That's
about
what it
the
looks
deep
like.
sea and
Yeah.
how the Earth works is always
Wow,
exciting.
that
That
looks-
might
Have
be
to
what's
tell
left
the
behind of
explorers.
Yeah.
one of these irregular
Yeah,
urchins.
urchins, it looks
Uh-huh.
dead.
Has Hercules ever
Sorry to
taken a
interrupt you.
test?
It's okay, that was cool.
Go ahead and come right there a bit.
I was just
And
saying,
passed with flying colors.
deep sea exploration and learning about how the Earth works is just so fundamentally awesome and exciting in itself.
It is.
Yeah.
It
It
shouldn't
is.
need further explanation.
But it helps.
It does help. It's just it's a process that we really are starting to reach the tip of the iceberg to understand and really recognize is probably pretty commonly occurring in the seafloor.
So,
That's fine.
40
Fine,
years
fine.
ago,
That's the
people
point.
were probably stating the same concerns about plate tectonics.
What do we make it now?
Why do we need to know about plate tectonics? Well, it's really what drives crustal movement,
Sure, yeah.
earthquakes, everything.
And just so you know, guys, science team, there is, you're getting a lot of plugs for the fluids. We've got, I think it is super exciting. Yay, fluids. Trust
yeah,
me.
not of this live nation.
Trust me, we get lit for discharging fluids.
That is awesome. And I wasn't saying it wasn't interesting. I just wanted to give
Put me
Annie
on the
I
spot.
wanted to give you a chance to
Yeah,
get people
yeah,
excited
yeah.
about what excites you.
Yes. And one of the things that really excites me is navigation and mapping and questions associated with those. So I'm going to throw this over to Aaron.
Woo woo.
And someone out in Nautilus Live Nation would like to know how far you've gone when traveling distance with the ROV. I assume GPS, etc., is useless.
Yeah, for our position on the bottom, we use USBL, so acoustic transponders, to know where the vehicles are, and then based on what the ship GPS is, we can figure out position of the vehicles. Once we get down, we also have
Zoom on in there on this guy.
dead reckoning using our Doppler. But we've covered lots of grounds with the ROVs, depending on what kind of operations we're trying to do. We've had Argus only doing side scan for, I don't know, felt like miles and miles. Not really sure.
Very
go
cool.
ahead, come
Thank you.
And, you know, just because we're ending the dives at midnight doesn't mean everything ends. We'll be mapping our way in. So if you guys are lonely, feel free to join me in the data lab. Very exciting mapping watch.
I am there.
I won't hold you to that.
Video, and go ahead
No,
and
I'm
zoom.
serious. I love maps. And I've learned so much about mapping through this. It's awesome.
Cool.
Right up there with discharging fluids.
That's one of them there grenadiers, right?
Looks like it.
Yeah.
Mhm.
Go ahead and come wide. Nice.
And I kind of heard the science team talking a little bit about what the fate of the whale bones will be when we get them up. Somebody was asking, how will you process them? Do they get frozen or?
We collected a number of different
You're on SDL.
What's that?
Oh, I can't hear.
We collected a number of different sample types, and we have been in contact with the different researchers who are interested in them or who requested them on how to preserve them best for their studies. Some folks have requested ethanol, which will help preserve for I think DNA studies, and also some folks have requested freezing for some of the other samples, and we might be subdividing a few different samples to share. Annie could probably talk more about how we process some of the squeezer samples.
Yeah, the squeezer samples, so they go to 2 different scientists. Typically, we'd ideally have collected the vent fluids, but we collected whale fall fluids. In a normal instance, I would filter vent fluids through a membrane for a biologist who's going to just take the filter and analyze what is left on it after the water goes through it. And then the geochemistry samples we collect for nutrients, we collect for metals and major and minor metals. Just a general trying to understand what components and what elements are in the water column, both chemically and biologically, and trying to relate that to reactions that would have happened in the flow path between the recharge and the discharge site.
Or between the whale and the water sample.
Yes.
And then maybe Magda, could you talk about how that
Go ahead and zoom here.
the Niskin samples are processed?
Yeah, the Niskin bottles, we transfer all of that water into, we have these sealed, they're kind of, they're like bladders that you would take like hiking with you, like to carry water. So they're like that, they're just sealed. So we'll put the water in there, and then we have a pump that will pump, like to filter the water through. It goes over a very fine like paper filter. So we'll pour the water into this pumping system. There's a jar attached to a pump, and you'll keep pouring the water. It pumps the fluid through, and then there's a fine filter that collects all the particulate and the DNA that's in the water, and then we basically preserve the filter. I'm not sure what solution it is in. Everything is prepared, pre-prepared, and sent to us. So, and then we preserve it and we mail it back to the labs that are interested in processing the eDNA.
Go ahead and zoom on there, Ashley.
Man, the squat lobsters down here are just huge.
Yeah.
Yep.
Yeah, really big.
Look at you.
It's a pretty neat feature of the management zone.
Gigantic squat lobster.
Thank you for that zoom.
And Magda was just talking about how to collect that sample for environmental DNA or eDNA, which is the DNA that is in the water, but you can't see it because it's kind of free floating after it's been shed from different organisms. And we presume that we're going to detect the DNA from the organisms that are in the general vicinity of the water that you collect or have recently passed through that water.
I wonder if the Niskin that we collected near the whale fall, when they analyze it for eDNA, if the DNA from the whale is such a high concentration of the sample that you aren't able to really recognize other accurate representations of the other organisms that are there.
Swamp everything else out.
Well, most of the time, my understanding with eDNA is it's not yet very good at talking about relative abundance of things, just more presence absence.
Okay.
So I think you can get a general sense of like if there's an abundance of one kind of DNA, but you can't do very good comparative metrics.
Do you know what kind of instrumentation they use to analyze the eDNA?
It's only dust down
Well,
to 2 as
it's
well.
on the filter, as she was talking about, and then it's extracted from the filter, and some primers are used. It depends what organisms they're looking for, how they prepare it, but then they're going to sequence it,
Mike, you want to come down a little?
and then they'll
get an
compare those
image of the sea
sequences
star?
to known
What's that?
sequences.
I'm going to come down a little on the winch, and I'll get an
Such
image
as
of the
those
It's only like 20
contained-
meters off the bottom. Come up. Just come up
I'm
slope.
trying to remember. There's a. I think you blasted against a library, or I'm trying to remember
Yeah, I
the
think-
terminology. Is that right?
I don't know the terminology, but I know you can compare it with
Yeah.
We can get
a
it.
database.
No, yeah, I'm trying to remember. It might come to me, the terminology for that. But
That's fine. I'll just get a picture of this guy. Video, gonna zoom in there?
I think on our website on Nautilus Live we have
Thank you.
eDNA, like a blog post explaining how eDNA works. So for any of our readers who are curious,
great. There's more information on environmental DNA there.
We haven't talked much today
Okay,
about
mind the
the POP samples. That sampling concluded this morning about 2:30 in the morning with the completion of our pumping. We pumped approximately 400 liters of water through special sampling media that we have on board Hercules, and those media will be returned to the chemistry laboratory and processed for concentrations of persistent organic pollutants such as DDTs, DDDs, dieldrin, PCBs, and flame retardants, PBDEs, so that we can know what the concentrations of those are in the waters around Davidson Seamount
What do you want to push it?
What is that?
Could you zoom in on that black thing?
help us answer other questions about
Plastic.
what we've been seeing
Oh, that's
in
plastic.
surface waters.
It is plastic.
is that a
You think it's a watch? I think it was some sort of a clamp, like that goes around a.
Like
Yeah.
a
Oh,
Velcro.
yeah, yep.
Yeah.
Actually, you know what it looks like
Thank
to
you.
me? It looks like the little plastic piece that goes in a watch case when you buy it, that the
Yeah,
strap
Oh,
goes
yeah.
that's
around.
right, that's right.
Excellent eye. Thanks for
Dang.
that zoom.
Yeah, sure.
yeah.
Go ahead. Yep, thank you.
It's definitely in the category of marine debris.
And another giant squat lobster.
Yep.
Yep.
So we have a question out there about
Can you
the
go ahead and zoom on this guy?
geologic history of Davidson Seamount, when the last time
That's great.
it erupted. I was trying to look online to see if I could find some information about that, but haven't come across anything. Science team, do you know anything about that?
Give me a second.
While she's
need
looking
to-
that up, I can.
Jen, no, Jen, move your mouthpiece.
I was just going to say that the outcrop is 9.8 million years old,
Yeah.
as far as the aged
All right, good.
basalt
Right there.
that they've looked at.
Thanks, Ashley.
Based on looking, when we were at the actual outcrop itself, looking at the rocks, it looks like there's multiple flows where you can see one type of rock kind of shape, and then there might be like a more sheety flow looking type thing over it, which is suggestive that perhaps it built up over time all around that same 9.8 million year time period.
Yeah.
Very cool, thank you.
That's all. But that's just the outskirt. That's one very small feature at the edge of Davidson Seamount. Davidson Seamount is actually a much larger feature.
So the pinnacle is here, or the pinnacle
Right?
is
Do you know the
over there?
geological history of it?
Well, so Erica Burton has just entered
Little
the
louder,
control
Jen.
van. Erica Burton has just entered the control van. She's going to be taking
Awesome,
over for
thank
me
you.
as watch lead on the next watch, and she has quite a background on Davidson Seamount, helped put together the information on it for the sanctuary's website, etc. So I am going to let her take over some of these more specific questions about its history. But that'll take a few minutes as we just transition over, and I can let her know what we accomplished on this watch and the plan that we for the next watch.
All
so
right.
anyway, thanks everyone for tuning in. I hope you enjoyed all that whale fall video and that we just collected and shared with you. We sure had a
Ooh.
great time.
That's a bright one.
What
Can you
an
zoom in on that
adventure.
pretty
Oh,
sea
that's
star?
Bye, Jennifer.
Oh yeah,
Thank
look
you.
at those.
Hi, and this is my last watch, so bye, Nautilus Nation. See you hopefully next year. I'm going to keep my fingers crossed that I get to do this again.
Oh,
Yeah, thank you.
Thanks,
Jen.
Jennifer. It's
Bye,
been a
Jen.
blast.
Thanks, Jen.
thanks for that zoom.
Of course. Something colorful.
Yeah.
Yeah, I've lost track of the time. It is just about time for a shift change.
Yeah, you're gonna start to hear buzzing in the van now as the new crew comes in. Thanks for that zoom.
Sure. Gonna come a little wide there.
This has been an amazing experience. I want to thank everybody. A great team of people. Thank you, Ashley.
Thank you, Scott.
You're welcome,
Yeah,
Mike.
thanks, Scott.
Thanks, Jess. Aaron.
guys.
I will see you in the mapping room, or in the data room. Magda, thank you so much for all the hard work that you've done and all the cool information. Annie, are you still there?
I'm still here, Scott.
Hey, thank you so much for all the awesome science and information that you've shared with us and the Nautilus Live Nation.
This has been very fun. Thank
Did-
you for being such a good communication fellow.
Oh,
Yeah.
you're
Keeping
very
us
welcome.
connected.
God, it's
And
been a
all the
blast. I'm like,
wonderful puns and jokes.
thanks. And like Jennifer said, I hope I get to do this again. I do this all the time. Dane, are you still there?
I am.
Yeah, Dane, it's great meeting you and fun
Same to
working with you.
Same to you, Scott.
You are a font of knowledge.
Well, yeah, you've carried us along on this watch quite well. Thank you, Scott.
Thank you very much. And there she is, all a yawn, Danny Bailey. Oh my gosh, I think you're, it's gonna be your last watch too, huh?
Oh.
Oh,
Well,
wow.
Thanks, guys. That was a good watch.
all right, Nautilus Live Nation, this is Science Communication Fellow Scott Emerson Simon signing out. Thank you all for joining in the adventure.
Excellent. My replacement is here, so I will say good night to you, Nautilus Nation. And here's Amanda.
Okay, Ashley signing off as well. Dwayne, bye Nautilus Nation. Thanks.
Okay, 4 to 8. All right. So how many viewers have we lost? Yeah. There's a lot of people still out there. Thank you for sticking with us. Don't know what we're going to see to top that, though, but we will try.
All right, good evening. This is Erica Burton sitting in the science seat from the Monterey Bay National Marine Sanctuary. We are en route to some slumping areas and then a cone area that we hope to characterize in the next couple of hours.
And in science chair number 2, my name is Amanda Kahn, and I'm from Moss Landing Marine Laboratories.
And this is Taylor Ann Smith, the data logger. I'm the ocean science intern, and I'm a master's student at Cal State Northridge.
Miles Saunders, the navigator for 48 Watch.
Dan, Herc pilot.
Antonella, Argus pilot.
And Wayne, your video engineer.
And this is Danny, your science communication fellow. Thank you guys so much for staying with us. We've got a lot of viewers going on. That whale fall was incredibly exciting. But we are here and exploring, and if you guys have any questions, send them on in to Nautilus Live, and we're here to answer them. Let's go find something else super cool. I can't wait.
So the pinnacle is, it's on our bathy map, is it? Or
Should be.
It's off the screen right now.
I think he's referring to something else.
Oh,
the
okay.
seamount, or whatever we want to call this thing, is most certainly on our map.
but the one you want to investigate is the one that's on the map there?
Exactly, yes. So we're going to make a pretty fast track over there, sort of passing by waypoint 16 and then onto the knoll to do a little bit more characterization in depth, up and down the knoll.
Okay.
Or around it, however you want to fly it.
How far away is it?
Uh, 1,300 meters.
It's an hour away at point, well, over an hour away at 0.5.
Yeah, if we could do 0.5.
And we'll be sightseeing along the way.
Yes, guys, I love the enthusiasm already. All of our internet explorers are ready to go. Not the browser window, the awesome people on the other side that are watching.
Can't hear you.
Yeah, sorry about that. Yeah, all the folks out there want to know what Erica and Amanda are going to do to top the whale fall.
I don't know if we can.
Dude, there's a sponge right there, guys.
Okay, we did it.
Holy cow.
Enough said.
So I might be biased. I study sponges and other suspension feeders, and these sponges that we're seeing down here are glass sponges, which I think are super, super interesting because they're super bizarre. So we're looking at what's likely Saccocalyx right there. It's a sponge that has this big stalk that projects it off of the wall of this clay ridge, and it has this big body up above, and it does that to grow up away from the seafloor to be up higher out of those sort of boundary currents that exist right down near the sediment, which is nice and slow. So up there it can get more food because more water passes by it. And why are glass sponges super cool? Well, like their name suggests, they make their skeletons out of glass. And they get that glass by extracting silica and oxygen from the seawater and depositing it into these rods and crosses and weird shapes. And so for you guys who are watching online, you already have a browser open. If you go to Google Images and write hexactinellid sponge spicules, you'll see some amazing shapes and forms. They're beautiful. They're also really neat because of how ecologically important they are. So they filter feed and they pump lots of water through their bodies, and they can extract bacteria from that water. And that bacteria, well, that changes the water in itself, but also the bacteria that they eat turns into either more sponge or into sponge poop that gets pooped out and deposited away where it's edible by other animals.
I'm noticing that this stock is very clean, and you were saying on a different species you can tell between, is it Caulophacus? You can tell if one's a dirty stock or a clean stock?
Yeah,
is that true?
Hyalonema, yeah.
Oh, sorry, Hyalonema. But this is not the one. This is a different one.
Yes, so this is not a Hyalonema because Hyalonema's stalk would be uncoated spicules. This one has tissue going all the way down the spicules.
And so it would catch a lot of more sediment and debris coming by.
Exactly.
Right.
That's good to know.
Yeah.
It is Amanda's birthday today, so happy birthday.
Yes.
Happy birthday, Amanda.
Happy birthday.
Sponges like the candle on the icing of the cake, which
There you
was a
go.
whale fall. It's been an amazing birthday. Holy cow.
So I'm looking up these sponge spicules, and there is no way I am messing with any of these glass sponges. These look like cool, dangerous ninja weapons.
Yeah, when we handle them on deck, we wear gloves. So
Yeah.
Do
Yeah, and I don't even use my hands. I use utensils to pick them up just to be safe.
just in case.
You know what? I would do
You
that.
have
I use my
chopsticks of
claw.
science?
Yes.
Chopsticks of
More like a
science.
huge tweezer set like pair, but that's pretty close.
Guys, who's ready for the super smelly whale parts later in the lab
Oh,
at
this is
like
gonna be
1 in the
fun.
morning?
Oh yeah, I'm ready.
Just
I'm
me?
typing up this dive report, and it's gonna be a fun time.
Yeah.
I want my own chopsticks of science.
I would like nose plugs of science.
Nose plugs of science.
But I like how forward-thinking you guys all are. I just got so excited when sponges would come up from ROV dives that several times I would run in and just grab them, smell them. They have interesting smells. And then I'd end up with those little spicules in my fingers. It's kind of like if you ever handle fiberglass. You get all those little rods and kind of itchy and kind of painful. So don't be like me. Don't be like past me. Present me has now finally learned.
Learning things.
Wow, this is quite the clay cliff. This is neat. We were looking at clay ridges like this earlier, but they were kind of smaller steps. This one is a nice big wall.
It is.
Yeah, can we tell how tall this wall is?
Just trying to look at the bathy map there, and then I see 5 or 6 lines really close together and warping in 12 meters
Wow.
below us at the moment.
Okay.
Oh. We've got
Okay, that's all I need to see. Thanks.
We've got lots of people writing in, wishing you happy birthday, Amanda.
Oh gosh, thanks everyone.
Very excited. Hi, Bobby's mom. Thanks for joining us. We theorize that sperm whales and giant squids have these epic undersea battles. Have there been any research ideas, expeditions to try and document these fights? Well, I don't know about fights, but there are scars from giant squids on sperm whales' faces, and there are giant squid beaks in sperm whales' stomachs. So there's a battle that's happening, and someone's winning.
It's a battle to the death.
It is a battle to the death, because someone either gets hungry and starves, or someone is eaten. I did have a question for you, Amanda. Are filter feeding sponges affected by microplastics?
Short answer, yes.
Yes.
Longer answer, more research is needed. So I don't know. I haven't looked in a couple of months, but I have actually been looking into this a bit. I don't know about studies that have looked directly at sponges feeding on or accumulating microplastics, but all suspension feeders are susceptible to gathering up microplastics. And I do know that for some of my research, I would feed microplastics to sponges to look at the way that their feeding happens and to try to find out what sponge poop looks like, because I could track those particles all the way through and out the other end. And so I do know that they took up the beads, and then they do poop them out, seemingly undigested. So yes, they eat them. Does it harm them? Don't know yet. We need to have some more research for that.
Huh.
And
That's
just
interesting. I'm
yeah.
glad it doesn't, like, I guess, get like super stuck in them.
Well, some doesn't. Some makes it through. It probably depends on the size. But yeah, somebody should really study that. Ooh.
They should. And as for the joke, how much deeper would the ocean be without sponges?
That's a profound question.
They are filling up
Sorry,
the
Wayne.
seas.
I promised I wouldn't do too many puns, but Profound.
That's okay. You fed me.
It has to happen.
I do want to expand on the suspension feeders with microplastics a little bit because I read this interesting study that surveyed mussels from supermarkets in the United Kingdom. They sampled different supermarkets and bought mussels, and they looked at how many of them had microplastics. And I think 7 out of 9 stores had mussels that had microplastics in their gut. So we should definitely know that plastics in the ocean are harmful, or they can travel. They can travel into animals in the ocean, into sediments, and then possibly back to us.
So we're eating those microplastics when
Yes,
we eat our shellfish.
If we have a chipino, yep.
And with our fish too, we're eating a lot of plastic, and plastic can hold on to a lot of pollutants and dangerous material, not only for the fish and the sponge and everybody else, but for us too. All of those compounds are not good for our physiology and our body system. So guys, we got to work on this whole plastic thing.
Well, the thing that's particularly troubling about shellfish is that the microplastics are in the part of the animal that we eat, because we eat the whole body. Fish, I'm not so sure that the microplastics make it into the muscle of the fish.
Yeah,
but
so
the
that
pollutants on the surface probably get somewhat extracted and incorporated into the fish's body.
They do. They do. Depending on the microplastic, it can be, or the toxin on the microplastic, it can be absorbed sort of through the lipid content of the fish, and then it can either be stored in the fat or it can be stored not the blood. It's sort of transformed into something else, and my mind has gone blank. But yes, we may not. I guess sometimes we're not eating the plastics directly, but we're getting the effects of that.
Yep.
Absolutely. Oh, Amanda, I have questions on questions on questions.
For me? For all of us?
For you.
Oh.
Okay.
Yeah.
Okay.
So what is the best smelling sponge?
Oh man, I don't know the species of it yet, but the best smelling sponge is this glass sponge that we brought up from Sur Ridge last year, and pulled it up, took a sniff, and it was sweet almost, and I couldn't quite place it, and so we sort of passed it around and had everyone take a guess, and then Jim Barry's the one who figured out what that smell was, and it was the smell of freshly cut lawn, freshly cut grass.
I love that smell.
It was lovely.
That is one of my favorite smells.
So what does that sponge look like so we can look out for
yeah,
it?
I want to
next
Google
time?
it.
It's white like a big sheet, and it has